Hi everyone, today we're going to discuss the interesting workflows of cell culture and we're going to highlight 2D cell culture as well as what's happening in the novel field of 3D cell culture. And to help me facilitate this discussion, Holly's on, and Holly, welcome.
Hi, Jeff. Thank you for having me. As Jeff mentioned, we're going to be discussing cell culture workflows. And to start this conversation off, we're going to start with 2D cell culture at the center of these workflows. Some of the workflows we'll be discussing today are 3D cell models, cancer research, drug discovery and development, and vaccine development. So let's get into it. We'll start off with 2D cell culture. So what is 2D cell culture? This is a very general and very conventional approach that has been used by most research scientists since the 1900s. It's the process of being able to nourish and grow monolayers of cells on a flat surface, be it at the bottom of a petri dish, a flask, or in growths in suspension. The general processes of 2D cell culture starts with the culturing phase, where we grow the monolayer of cell in suspension for non-adherent cells or in tissue culture flasks. The nourishment for these cells is obtained from growth medium or from serums. The next stage, after we've grown substantial growth of cells, we can then plate these cells or alicate them and plate them into 96-well plates, 6-well plates, and then use this for further treatments and analysis. The process of being able to do cell plating is by counting the cells. We can do this in two approaches. There's the automated approach using automated counter, or the manual approach, which is using a microscope and a hemocytometer. There are different treatments that can be used on these cells in plates or in any kind of medium that you choose. The treatments are usually using compounds or reagents. There are different types of compounds and reagents, and it's dependent on the research question that a research scientist is trying to ask and what they're trying to achieve or observe. For instance, transfection reagents can be used on culture cells to insert modified DNA into the cells for expression of proteins of interest. Once we have our treatments and experimental designs completed, we can then look at cellular imaging and doing the analysis of our cells. And so within cellular imaging and cell imaging and analysis, we can use very many different approaches, some cell-based assay approaches, such as looking at cytotoxicity, cell viability. We can use microscopy or cellular imaging systems to observe different markers or stains, such as fluorescent proteins, to then monitor protein interactions or to be able to monitor transfection efficiency. Imaging can be done manually, like through microscopes. It can be automated through microscopy or cellular imaging systems, and it could also be high throughput if you have a lab that has a large amount of samples that you're running.
I want to find out, you mentioned earlier culturing. I mean, I understand that sometimes people obviously pull from a cell bank or they're looking at commercial cell lines, especially in the therapeutic space. Do we offer support in that culturing space as well?
Yes, we do. We also offer cell lines from different suppliers, dependent. It can be general cell lines, primary cell lines, as well as specialized cell lines, including knockout genes, which becomes more applicable in research such as, cancer research.
Okay, awesome.
If we move forward to the next stage, if we look at a product overview of some of the offerings that we have for 2D cell culture, we're able to offer media from Cytiva Hyclone. This can be classic media, specialized media, and the serums such as FBS or donor equine serum. We offer all the plasticware that's needed for tissue culture. This can be tissue culture flasks, the pipetter, serological pipettes, or test plates. We're also able to offer biosafety tube cabinets, CO2 incubators, or centrifuges. We're also able to offer a wide range of microscopy platforms, from inverted microscopes to fluorescence microscopes. This can be from routine, wanting to look at your cells, doing cell counting, just wanting to observe your cells, all the way up into confocal, wanting to actually look at markers, wanting to do 2D or 3D kind of analysis on your samples. We then offer more automated systems, molecular devices. They can be cellular imaging systems or high content screening systems. When it comes to cell-based assays, we're able to offer microplate readers. This is everything from absorbance, fluorescence, luminescence, TRF, alpha screening, you name it. So these are kind of the instruments and the media and products that we offer that can help you get your cell culture lab started up and running.
Awesome, excellent. So I see here some bioreactors, so I'll maybe jump in on this slide. I mean, often what we see is, even at a university level, but also commercially, when someone starts to move away from tissue cultures, it's just very laborious to have a stack of flasks in your incubators, plus you need multiple incubators. So we start to move towards this framework of looking at bioreactors, whether that's at the bench scale all the way to fully, 2,000 liter bioreactors or fermenters. And a big decision maker right there in the front is, are we dealing with adherent cell lines or suspension cell lines? The nice thing is we do have certain technologies that are specifically catering for adherent cell lines, like the iCELLis, that really focus on a whole bunch of membranes that are kind of woven together to allow for, just high density cell culture. Additionally, when we deal with some adherent stem cells, we have things like the Xpansion that really focuses on these plated bioreactors. And you can have up to 200 different plates in these bioreactors. It's almost like a tower of sorts that has a central column that feeds those plates. And that's quite a nice technology. But often what is the most scalable is when you can move products or cell lines from adherence to suspension. And when they're in suspension, we can really start to do a lot in terms of just the mass of cells that can be grown out by these different bioreactors. Now, we typically need a seed train when we seed from tissue culture flask to a smaller bioreactor and then a much larger commercial bioreactor. And often questions that come in is like, where's the GMP, which is good manufacturing practices. So whenever you're producing a therapeutic or a certain high value product that's going to be going into definitely clinical trials, you're going to need to demonstrate control of the manufacturing, which does require good manufacturing principles to be applied to the manufacture of that equipment and the entire quality management system for that. And that's often where we tie in with the research side of Holly on, that development and transition into that whole world of GXP products associated to the respective regulatory requirements. And you would have noticed there we have suppliers from Applikon to Pall, we have single-use fermenters from Cytiva, and we do an array. I mean, we've mentioned the medias, but we also do a lot of peptones that's often used in the veterinary side for E. coli fermentation. So with that, Holly, I'm going to hand back to you because we're going to go into that interesting world of 3D cell culture, and that's really an interesting space at the moment.
Thanks, Jeff. Okay, from 2D cell culture, we can move on to 3D cell models. And why 3D cell models? So 3D dimensional models have become more physiologically relevant. They closely represent tissue microenvironments, cell-to-cell interactions, biological processes that naturally occur in vivo. There are many different approaches to being able to create 3D models. One of the approaches that we can look at is scaffold-free. What this means is that we're able to culture 3D models without requiring adherence or 3D dimensional material such as support matrix or being able to seed our cells. Examples of this are things like ultra-low binding plates that are coated with hydrophilic polymers. This prevents cells from sticking to the surface and allowing clusters to form and for them to be able to form their own extracellular matrix. We also then have engineering-based approaches to be able to create 3D cell models. These approaches include 3D bioprinting. This can be in the form of being able to create that extracellular matrix or support structure to assist with cells being able to adhere to form their own 3D structures. We can also use 3D bioprinting with cell-laden hydrogels or bioinks to then directly create a 3D bioconstruct. There are also other approaches that we look at 3D cell models, which include microfluidics. This is a very interesting field. And this is such things as organ on a chip, which is a 3D microfluidic circuit, integrated circuit. They're basically miniature tissues that are grown inside fluidic chips. And these devices mimic human physiologies and allow you to control the microenvironment. These 3D cell models, such as spheroids and organoids, can be treated with different compounds or reagents. And then we use 3D imaging and analysis to then produce more physiologically relevant information. We can use 3D confocal images or imaging systems to not only observe the X and the Y, but also the axis of our sample. And this produces a lot more physiological relevant information. We can produce high resolution imaging of these 3D models using different approaches in microscopy, such as digital light sheet, which you can do and achieve using the Leica confocal system. 3D models have become very important and relevant in many other cell culture workflows, such as drug discovery and in cancer research.
Awesome. So I'm going to jump in there. I mean, the thing that I find the most interesting about spheroids is specifically like its clinical use. and especially as drug development. When we talk about phase one, I mean, there's often a requirement for animal models. That's still the regulatory requirement, but there is a strong push by multiple researchers in the clinical space to try and establish the spheroid model as almost a preferential next step in terms of potentially circumventing animal models. It is still a requirement from a regulatory standpoint. However, there is some openness from regulators globally to start considering the viability of these models for more accurate in vitro modeling that correlates to a large degree to what they expect in vivo. It will be some time before we see animal models maybe being removed. There are some exemptions, when it's a very specific type of model that is being, or drug development cycle. But it's an interesting space. I mean, it'd be great to see that this just gets better and better, the technology, and minimizes the risk to patients when they model in these new candidates for clinical work. And then the other thing you mentioned was 3D cell printing. I mean, this is super interesting. Is this happening in South Africa? Are we seeing 3D printing applications happening?
It's growing. Yes, we are. We're absolutely seeing a growth in 3D bioprinting in South Africa. We actually have placed a system recently. And I think that space is very exciting, and we're moving towards it. The technology is there. We're able to offer it. We're able to assist. We're able to integrate it into different types of workflows. So yeah, absolutely.
Okay, well, I'm going to encourage the South African researchers to make sure they put a lot of effort in there, and why not go for? I'd love a synthetic liver at some point. I might need one if we keep up the late nights. But definitely, it's awesome to see the technology taking pace, and the potential of even 3D organs for me is mind blowing to see what the future might hold. But Holly, I'll let you take us on to the next workflow.
Thanks, Jeff. So if we start looking at a product overview of what we can offer for 3D cell models, we'll start with the bioprinters. We represent a company called CellLink, and we are able to offer extrusion-based bioprinters or light-based bioprinters and biodispensers. Broad range of different systems, different throughputs. If you're in the entry level where you're just trying to do pilot scale studies all the way up to larger scale, we're able to assist with different systems. We're able to offer cell analyzers from Cytiva if you're looking to monitor, do live cell imaging of your spheroids or organoids. This is the system for you. You can look at cytotoxicity, cell viability, and just generally monitor the growth of your 3D model. We're also able to offer a broad range from Leica in terms of microscopy systems, 3D imaging systems, cellular confocal systems. This allows you to then do Z-stacks and then look at a full imaging of your entire 3D model. We're able to offer digital light sheet. This is high resolution of your 3D models. This is also applicable in many different aspects of 3D modeling, and also automated cellular imaging systems such as Tamika. We're also able to offer plate readers if you're doing any immuno-based assays or 3D cell model assays, cellular imaging systems, as well as high content screening systems. We're also able to offer 3D spheroid culture medium. So this is specialized media towards being able to develop and grow 3D spheroids in cell culture.
Okay, awesome. Looks like we're well set up for that space.
Yes, absolutely. And I think as the space is growing, we're moving, and more products are being moving towards developing and being able to increase the capacity of 3D cell models.
Awesome.
So from 3D cell models, we can move over to cancer research workflow. And this is the big space in where 3D cell models have also become more relevant. So if we look at cancer research, both 2D cell culture and 3D cell culture we find in the space. But spheroids and organoids and even tumoroids have become more relevant in doing research in cancer research. So the first stage is to then culture our spheroids. And as mentioned before, this could be done in ultra low attachment plates, round bottom plates, or any labware that's been developed to just typically morphology of a spheroid. Once we have formation and growth of our spheroid in culture, we can then treat it with desired compounds or reagents of interest. We can monitor the effectiveness by doing things and looking at things like cell viability and running assays on plate readers. Treatments can be done with reagents. We can then look at analysis and imaging of our spheroid images, where we can take images within the body of the spheroid. This can be captured individually, or we can run through an entire Z stack where the multiple images are taken at different depths of the spheroid. We can analyze our cancer cells using cellular imaging analysis software, where we can further run qualitative or quantitative analysis to monitor the expression of different markers or to quantify different biological readouts. The next stage that we can look at, or the next workflow that we can look at, is drug discovery and development. If we get into drug discovery and development, we have different stages that we go, and this is in the beginning stages before we get into clinical trials. This is where we find that cell culture, be it 2D or 3D cell culture, becomes a little bit more relevant. The first stage of drug discovery and development that we look at is target identification and validation. This is the stage of being able to actually identify biological molecular structures that we feel have a therapeutic effect. And then we conduct validation experiments to actually prove this therapeutic effect. In lead generation and refinement, it's basically to then sort out the main candidates found in a large library. And we then look at these candidates, which we call leads. And these go through various optimization stages into the development of the drug. The optimization stage is a very critical and crucial step to secure a very solid candidate during the drug discovery stage. So ways that which we can do this and do the lead optimization is through things like high throughput screening. High throughput screening is us being able to then test this compound library against a biological target. Screening can involve replicating thousands of biochemical reactions to test for 100,000 drug compound targets. We do this systematically using a wide range of concentrations. We then move on to preclinical studies after lead generation and refinement. And this is where we start to look at toxicology profiles of new drug candidates. This is prior to administration in animals or in humans, and also to further extend what is known on the profiles of existing drugs. The ultimate goal of preclinical studies is to accurately model the biological effect of a drug, predict the treatment efficacy and safety of the drug. Prior to administration in vivo, in vitro studies, as you've mentioned, Jeff, we're using 3D models such as spheroids and organoids. Over to 2D cell culture is becoming incredibly more relevant. It is a more integrated step in the drug discovery development process, and 3D models become a very important step before moving into the next stage of development. As we've said, it provides more physiological relevant information on new drugs, which seem to fall on the limitations of 2D cell culture. And this can result a lot of the time in failed drugs upstream of the drug development process. So 3D models, not only a step before in vivo, it's becoming just more relevant in the space to prevent, drug discovery and development is a very expensive process in the long run. And being able to validate drugs at the early stages is quite vital and quite important.
Yeah, so I'm going to jump in there. Anyone who's kind of looked at, the clinical side of it, it's really, really expensive to be taking a product through phase 1, 2, 3, and the respective phase 3 A and B if you need to, before even going to the final commercialization stages. So, for me, like these tools, it's quite comprehensive, but a lot of this is intended to cut down on that, make sure that your hit candidate is appropriate, and then obviously map that toxicology as thoroughly as possible and try to assess, is this going to be a blockbuster? Because what are the limitations, in terms of, it's nice if you're looking at certain organelles, being able to test those organelles for cytotoxicity, known modes of, how it's going to be processed throughout the body. It really does save quite a lot in the clinical development side. And it's definitely the combination of genomics, proteomics, or omics are kind of pooling together at an early stage with these candidates to just really scrap a poor candidate early in the process and then really just put investment behind those star blockbuster candidates that really show promise. And if we want to develop more of this in Africa, we definitely need a lot of these resources that I'm seeing on the screen here to enable these workflows for drug development. So awesome, Holly. Great overview.
Thanks, Jeff. So from drug discovery and development, we move on to vaccine development. Within this workflow, it can vary, and it's dependent on the platform that you use, whether it's inactivated viruses or DNA-based vaccines. So what we're going to do here is just kind of give a simple overview of a few virus-related workflows, from antigen to antibody discovery to cell line or stable cell line development. So let's get into it. We'll start off with antigen discovery. If we go into this workflow, where there are various different steps that we can move through. And the first step is to screen for binding to virus-specific immunogens. There are very powerful tools that we can use in this stage to display and screen for millions of antibodies against viral antigens. One of these is using phage display, for instance. Those with a high binding affinity will then be considered as potential vaccine target or candidates. Okay, the next stage we can look at is immunogen characterization. This is where we conduct immunogenicity studies. This is whether to determine if the identified antigen will then initiate immune response. If the antigen invokes an immune response, and we can also measure this with plate readers doing ELISAs, the antigen will now be considered an immunogen. So just to clarify this a little further, this is the main difference between the term antigen and the term immunogen. Not all antigens that we find in the first stage is capable of invoking an immune response, and only those that invoke immune response are known as an immunogen, and these candidates move forward in the process. So the third stage of this is to then do protein or immunogen engineering. There are some properties of immunogens that are known to have undesirable effects, and when injected into animals or humans, therefore we can use genetic tools like CRISPR-Cas9 to then manipulate DNA encoding the immunogen so that they no longer possess those properties. On the other end, we're also able to engineer desired properties into the immunogen for the next stage. Once our DNA has been optimized, we can then scale up the production of the immunogen for further testing. And a way that we can do this is by generating a high-expressing clonally-derived cell. In order to achieve this, we can perform cell transfections and single-cell cloning. Cloning or single-cell isolation is the process of being able to separate an individual living cell from the rest of tissues or group of cells. And this becomes incredibly relevant in this workflow. After we've done single-cell cloning, cell lines are then monitored for growth and assessed for their protein expression. This is the final step of antigen discovery. The immunogen is then purified, prepared for injections into animals or to induce an immune response. And the antibodies generated from the animals are then screened in virus neutralization assays to determine the vaccine efficacy. So this is the final stage of the antigen discovery. And next, we'll look at antibody discovery. And there are two mechanisms in which we're going to look at and cover today in antibody discovery. The first mechanism that we're going to look at is phage display. What is phage display? Well, it's a technique to be able to study the interactions of proteins displayed on the surface of a bacteriophage. This then interacts with molecules such as peptides, DNAs, and other proteins. We use phage display to find high affinity binding or interactions with antibodies and antigens, and this plays a crucial role in the vaccine development. The first stage that we look at is panning, and panning is an iterative process to then enrich for a specific bacteriophage in a large library or a population that shows a high binding affinity for the target. This is done by exposing the library of antigens of choice, eluting it out, amplifying it with only the phages that have the highest binding affinity. So once we have a bacteriophage selected in the panning stage, we can then move on to colony picking. This is where we pick and isolate specific colonies that show unique protein binders. This process can be fully automated, and higher success rate can be selected through colony picking using automated systems, and some of those systems that we can offer are things like the cube picks from molecular devices, and this is microbial colony picking. We can then, once we've done our picking stage and we've isolated our bacteriophage of interest, we can then do further antibody interactions where we validate using more quantitative immunoassays. This can be to quantitate the antibody-antigen interactions using things like ELISAs, immunofluorescence, HTRF. There are many different approaches, and we can use plate readers and cellular images to then monitor this approach. This then allows us to then look at the functional screening of our antibody or our antigen that we're looking at. And this is to then further characterize the antibody-antigen interaction. The candidate molecules are then screened for functional activity, and this is through the process of things like looking at viral neutralization and determining once again, the vaccine efficacy. We also often use cell-based assays to try monitor this and to try achieve this.
Okay, awesome.
Our next stage, or our second way of being able to look at antibody discovery, is through hybriderma screening, is a method for being able to mass produce antibodies in a hybrid cell line. The first stage of this, or the hybrid cell line, is generated from the fusion of antibody-producing B cells with an immortalized myeloma cell line, and that's what we call a hybriderma cell. Second stage is to identify and clone hybriderma cell lines, which are producing high amounts of monoclonal antibodies. The screening of successful clones can be done using things like a mammalian colony picker, and this can be a completely automated approach, and we can offer products like the clone picks for molecular devices.
Yeah. What's nice about this, sorry, I'm going to jump in here, is I've seen some of this monoclonality assurance that these devices apply to the selection. That's often the issue is, when you are screening these hybridomas, like really finding that specific high throughput candidate and not pulling through another cell that maybe is, giving you a conflicting signal. It's really quite high-tech technology from molecular devices. It's really impressive. Yeah.
Absolutely. And there are many validation steps that you can use to be able to monoclonality assurance specifically. And we do offer other systems from Cytiva with regards to being able to do the validation process of this as well. So once we have our clone of interest, we can monitor cell growth. And this can be done by monitoring and observing cell divisions over a given period of time, and we can use cellular imaging systems to be able to achieve this. The process of then being able to look at antibody interactions, we use this to then analyze the ability of our antibody to bind to only specifically one antigen. And this is where we then look at things like affinity and specificity. And then once again, we'll do functional screening, where we'll perform viral neutralization assays and then determine the efficacy of the vaccine itself.
Okay, awesome.
And once we've done antigen and antibody discovery, we look at things like cell line development. And if we jump into cell line development, we'll run through the different steps. One of the first steps of cell line development is to then look at stable transfection. So it begins with the introduction of foreign DNA, which is encoding your recombinant protein of interest into the host cell. And that process is known as a transfection. And upon transfection, the cell begins to transiently express your protein of interest for a certain period of time. This is generally often a week before it completely halts production. But there is a population or a subpopulation that maintain their ability to be able to produce that recombinant protein for a much longer period of time. And that's because it has been integrated into the genome of the cell. So those are then referred to as stably transfected cells. And that's what we want to enrich for in the next stage. And so the foreign DNA that's encoding the protein of interest, usually we can identify it with a selectable marker to be able to then pull out or enrich for that specific cell that we're looking for. And we can do this with things like fluorescent markers, for instance. So green fluorescent proteins are often added to the foreign DNA, and we can use that as a marker to then pick out or select for our specific colony. Also, there's a strong correlation, for instance, between things like the level of recombinant protein expression and the level of the GFP marker. So it allows the intensity of GFP to then enrich the pools for high protein expression, or be able to pull out for those that have a high protein expression. The next stage will then be to isolate that cell that we're looking for, so single cell isolation, in order to ensure that the population is genetically identical. Okay. Single cell cloning is an extremely critical stage of the cell line development process. And so post single cell isolation, we can then do monoclonal and verification and growth studies. It's important that we identify single cells that are properly isolated from one another. You can use this using a cellular imager, for instance, clonality assurance systems from Cytiva. Once cells are isolated, they can be monitored for growth, and we monitor the growth to track the progress of the single cell into colony cells or colonial cells, and to make sure in their growth that there have not been any dramatic changes. The final stage of vaccine development is that we can then look at titer and QC screening, and this is to test for the amount of recombinant protein or antibodies produced from the clonally derived cell line. And then that's the final stage of vaccine development.
It's definitely been quite thorough. So we've covered that entire spectrum, the antigen, the antibody, the cell line. So I see we have a lot of products here that support these awesome workflows in terms of the entire plethora of 2D and 3D cell culture. Holly, I mean, we've comprehensively covered this from the cell plastic side, the media side, whether that's Cytiva, there's Conda for the fermenters, the bioreactors, there's a range there from Pall, Applikon, Cytiva. It's really a plethora of products, cell clarification as well. So it really is quite a lot that separations can support and supply. And be sure to reach out to us to support you in these workflows. I mean, we've tackled cell culture already in this one. We've had previous discussions, a very nice one on protein workflows. So if you haven't seen that, please go have a look at the protein workflow coverage. And I'm sure at some point we also need to be talking about mRNA vaccines, because that's a very hot topic. A lot is happening in the space. You know, what we've discussed has been more traditional approaches towards vaccine manufacturing, but it really is an exciting world of seeing how African science is being increasingly enabled in the region. And Holly, thank you so much for your time and taking us through that very comprehensive overview of all the different approaches with 2D and 3D cell culture.
Thanks, Jeff. Thank you for having me.
Awesome.